everybody, I'm Louisa. I'm a PhD student with Amy Kuceyeski at Weill Cornell, and Amy couldn't be here with us today because of geopolitical funding issues, but yeah, I'm happy to be here with you all today. So I'm going to talk about our use of network control theory to better understand markers of substance use or vulnerability for substance use disorder. And I have no disclosures. So substance use disorder, or SUD, remains one of the most challenging public health issues to address. And in 2023, an astounding 16.5% of adults in the US met criteria for SUD. And yet there are very few effective treatments for the disorder. And this is both due to the complexity of the neurobiology of SUD as well as a sort of rich interplay of biosocial factors that are involved. And historically, our understanding of addiction has also been limited by two sort of myths around addiction on where to place the blame. The first is that it's simply a matter of an individual's poor choices and some sort of moral failure of willpower. And the second, that it's the drug's fault, that drugs are inherently and universally addictive. And both of these myths really oversimplify and stigmatize the lived realities of people with SUD. And in reality, what we know is SUD is a chronic relapsing brain disorder. And for someone who progresses into SUD, they typically go through three stages where they start with recreational use, which many of us partake in. And this then turns into intensified habitual use, and then a loss of control and full-blown addiction. And at the core, it's thought that this is caused by a dysregulation between top-down and bottom-up brain processes, where an impaired inhibitory system of prefrontal regions is no match for an overreactive reward system in the limbic areas. And it's this imbalance that's thought to kind of trap an individual in this cycle of drug use despite the severe consequences. And there's a very large body of work showing structural and functional correlates of SUD in the brain. And I'm just going to mention a few here. So there's abnormal dopaminergic signaling, impaired white matter integrity, reduced gray matter, altered functional connectivity, and altered network dynamics. And a lot of these findings have been localized to regions and networks involved in this top-down, bottom-up imbalance that I just spoke of. But what's important to consider is that not everyone will follow this trajectory. In fact, for most addictive substances, just around 20% of people who use them over a lifetime will develop dependence. And in recent years, there's been more of an effort to understand the brain features that are responsible for this vulnerability. And so what's been found is there's actually similar patterns in the brain of those who are predisposed to SUD as to those who actually have the disorder. So again, dopaminergic signaling, white matter integrity, gray matter volume, functional activity, and network dynamics in the similar networks and regions as in SUD itself. So this raises some critical questions for neuroscience. Which brain features associated with SUD actually reflect the neurotoxic effects of drugs compared to which are reflecting pre-existing vulnerability? And the importance of the vulnerability is exemplified by the fact that adolescents who are known to have a sort of developmental imbalance between bottom-up and top-down are known to also have a higher risk of SUD. This time lapse here is showing from ages 5 to 20 cortical gray matter reduction as a marker of maturation. And this starts earlier in the sensory and limbic regions and then moves on to higher order frontal areas. And so this supports the dual systems theory of adolescents having a higher SUD risk due to a stronger bottom-up drive and a impaired or underdeveloped top-down control. And so a key population for kind of teasing apart cause from consequence is then individuals with a family history of SUD. And that's what makes up the focus of my work. This group has up to 8-fold higher risk of SUD, and they tend to experience more severe outcomes when they do develop SUD, and that's due to both genetic and environmental factors. And this recent review compiled replicated findings in individuals with a family history of alcoholism. And many of these findings are again in these limbic and prefrontal regions and implicating this kind of top-down, bottom-up imbalance. but only 5 of the 48 studies included only restricted their cohort to those without any prior substance use disorder. So it's very hard to draw conclusions, especially what we know about the effect of drugs on developing brains. And then to shift gears slightly and just give a little more background that's relevant here, I want to mention another important aspect, which is the impact of sex and gender on SUD, risk, and manifestation. And so a growing body of work is showing that there's a divergence between males and females in terms of the effect of the reinforcing effects of drugs, where females seem to be more sensitive to negative reinforcement and males more sensitive to positive reinforcement. So in males, they tend to initiate drug use based on the pleasurable effects of drugs, peer use, whereas females tend to initiate drug use to cope with stress and comorbidities. They also have more severe withdrawal and craving, which tends to lead to faster escalation of drug use and higher rates of relapse in females. But there's a historical lack of consideration of these factors, and especially in developing populations and SUD predisposition. And today I'm going to be talking about sex as a biological variable. I'll treat it as a binary variable just due to the limited sex and gender diversity in our cohort, but we'd like to acknowledge that sex and gender both exist on a spectrum, and we hope to be more representative of that in the future. Okay, to kind of tie together all that background, I've said that SUD has these functional and structural phenotypes that suggest an imbalance of top-down and bottom-up processing. The adolescents, who we know are more at risk, showed this sort of imbalance as well. And then youth with a family history of substance use disorder have a more exacerbated risk and perhaps a more exacerbated imbalance between top-down and bottom-up. We know that sex shapes risk pathways in the manifestation of SUD, but what we don't know is which brain features reflect the cause versus the consequence of SUD and how that is modified by sex. And so here comes the network control theory. We used network control theory to study vulnerability of SUD in a substance-naive population of males and females with a family history. And as we've heard, network control theory models how the brain is able to transition between activity states based on the underlying structural connectivity, denoted as A here. And then the control input, U, represents the minimal energy required to steer the brain from one state to another, and we call this transition energy, or TE. And TE provides a way to kind of quantify the cognitive demand, if you like, of a transition. And given that we know that SUD risk is linked to both structure and functional changes, and this sort of imbalance in the input required of top-down versus bottom-up, we thought that this was a good framework to study our question. And just to say, we used a uniform control strategy, treating all regions equally in their ability to steer the system. And then this work was also inspired by a previous paper from my lab by Parker Singleton and Puneet Velidi, who had found that globally across the whole brain, in the HCP young adults data set, they saw young adults who had heavy alcohol use had greater global transition energy, and that was particularly true of transitions, sort of top-down transitions to lower order brain states. So here, the visual brain states. And so given all that background, what did we do? We used the ABCD study, the baseline visit, just substance-naive adolescents who were like 9 to 10 years old, and in a cohort just below 2,000 who survived quite strict quality control. We split this group into those with a family history versus those without. So FH plus, FH minus, where those with a family history, we considered them FH plus if they had 1 or more parents or 2 or more grandparents, and then FH minus had none of either. 2 or more parents or grandparents with a history of substance use disorder, not just having parents. And then for the data, we used resting state bold time series from all subjects and a group average structural connectome, which was from diffusion MRI and tactrography. We had an average from a subset of this cohort. And so when we're talking about network control theory, we're saying how much energy is required to transition between states. And the way that we define states in this project is using k-means clustering. So we threw together all the bold time series across subjects, put that into k-means, and defined commonly reoccurring brain states in this population. And so what we could then do is assign every time point of each individual's scan to one of these clustered states. And so here, for an example, this subject might start in state 3, move to state 2, to state 1, go to 4, maybe stay in 4, and so on. And so what we end up with is, for every subject, a vector of states that then describes the temporal trajectory of that scan. And so from that, we then can calculate subject-specific centroids for each brain state. And what we identified using an optimal K of 4 was these 4 brain states shown here. And then we use cosine similarity to kind of give them a label from the Yeo-7 networks. And what we end up with is states representing high and low amplitude activity in the DMN and the visual networks. And so now we have our brain states, we have this group average structural connectome, and then we can calculate the transition energies required between transitions between each of these pairs of states and within a given state. And we calculate these transition energies at 3 levels, the regional, network, and global. And for each of those levels, we have a pairwise TE, which is sort of a matrix of the energies required for every pairwise transition, and then a mean TE, which is the average across those transitions. So here we have 86 regions, we have a matrix per region and a mean TE per region. We can then sum regions within the Yeo-7 networks and a subcortical and cerebellar network to get a matrix and a mean TE per network, and then the same for global TEs where we summed across all regions. And I'm going to start with the results in the global TE. So what we found using ANCOVA models controlling for various covariates was that there was no effect of family history at the global level, but there was a significant effect of the interaction between sex and family history. And what we found was that there's an opposing direction of effect in males and females, where females with a family history had a higher global TE compared to males with a family history had a lower TE. And we found this to be true across all transitions. So there's a decrease across all transitions in males with a family history and increase in females. And the effects here weren't super strong, so we wanted to then look at what's going on in the network level. And here are those results. You can see in A that there's, for ANCOVA models on network TE, we found no network had a significant effect of family history alone, but in the interaction between sex and family history, we see the default and ventral attentional networks, as well as the default mode network, had significant effects, where the ventral attention didn't survive correction. And when we looked into sex comparisons, we see that the default mode effect is driven by an increase in TE in the default mode network in females with a family history. And then in the dorsal and ventral attentional networks, this effect was driven by a decrease in transition energy in these networks in males. And this is evident here, where we see in the default mode network, we only see significant effects of family history on pairwise TE in the default mode in females, and then males showed the same, only males showed effects in the opposite direction in the attentional networks. We then looked at the regional level, where we found that across both males and females, there were some regions, specifically in the somatomotor network, that showed greater TE in both males and females. And then for regions that showed significant sex by family history interactions, it seemed that they were primarily driven by one sex or the other. And this sort of recapitulated our network findings, where default mode network regions were higher in females with a family history, and dorsal and ventral attentional regions were lower in males with family history. And so to kind of tie this all back together to the background I went over, here I'm showing again how adolescents are thought to be especially vulnerable to SUD because of this kind of lag in the functional development of the inhibitory control system. We think perhaps that in family history of substance use disorder, this is true in distinct halves for females and males, where now I'm showing lower TE as you go up the y-axis. And it seems that perhaps in males, the lower TE in the attentional networks may suggest a reduced energetic barrier to reward-driven bottom-up processing. Meanwhile, family history positive females and their greater transition energies in the default mode network may suggest greater energetic demands for more higher order top-down inhibitory control. And then to contextualize this in what's known about sex differences, it seems that family history might amplify sex differences by widening this gap. So females with a family history may be even more sensitive to the negative reinforcers of substance use, potentially due to a dysregulation in the default mode network, which has been associated with craving and withdrawal symptoms. And then on the other hand, this decrease in the attentional networks, TE, in males with a family history may show a sort of disinhibited dynamics in the attentional systems, which may heighten then the positive reinforcing effects of drugs. And again, this isn't a strict binary. There's no male brain and female brain, but importantly, geopolitically importantly, if we had ignored the interaction of sex, we wouldn't have found a effect here of family history. So while there's definitely individual variability between people, when you're looking at population level effects, it's important to consider biological sex. So yeah, in summary, we find that family history of SUD shapes brain dynamics in opposing ways. And the metaphor here is females have a harder time stepping on the brakes, and males have an easier time stepping on the gas. And then we think that this is potentially important for improving prevention and treatment strategies in SUD. And I'd like to thank the organizers for having me and my research team. This will hopefully be published soon in Nature Mental Health, and you can come find me tonight at the poster reception. I'll be bopping around. Thank you.